Hello, my name is Miwa, and today I'll be talking about the pancreas, gallbladder, and the biliary ducts. Before actually getting into the topic, let me explain how the highlights in my slides work. Red highlights are for terminologies that you must know for the exam. Orange is for terms recommended to know for ace in the exam, though not essential. And green is for terms that you do not need to memorize, but just good to keep in mind. First, let's talk about the structure of the pancreas. We'll start with the ampulla of Fata, which is an opening where pancreatic secretions are drained into the duodenum. From there, we move to the main pancreatic duct. This branches into the interlobular ducts, then into the intralobular ducts, and finally into the intercalated ducts, which terminate in the lobules as centroacinar cells. These centroacinar cells are surrounded by acinar cells. Now, if we zoom in on the lobules, you'll also find Langerhans islets. This shows that the pancreas is a both exocrine and endocrine gland, with the acini for the exocrine function and Langerhans islets for the endocrine function. The Langerhans islets are really important, both functionally and histologically, but we'll cover that in our lesson on the endocrine system. By the way, if the structure of the pancreas reminds you of the salivary glands, you're on the right track. But there are key differences. In the pancreas, there are no myoepithelial cells surrounding each acinus, and there are no striated ducts, unlike in the salivary glands. Next, let's focus on each cell in the pancreas. Acinar cells secrete digestive enzymes, including trypsinogen, pancreatic amylase, pancreatic lipase, deoxyribonuclease, and ribonuclease. You'll also learn other enzymes in your physiology lessons. The centroacinar cells secrete bicarbonate, which neutralizes the acidic chyme coming from the stomach. The cells in the intercalated ducts are cuboidal and they also secrete bicarbonate. As for the cells in the intralobular and interlobular ducts, they are columnar. Now, the pancreas secretes enzymes that digest sugars, proteins, lipids, and nucleic acids. But how does the pancreas itself avoid being digested? First, the activation of zymogens into active enzymes only happens when they reach the duodenum. Second, trypsin has a trypsin inhibitor. This means that even if trypsin is produced in the pancreas, it's immediately suppressed by trypsinogen there. Lastly, bicarbonate keeps the zymogens inactive. Next, how is pancreatic secretion regulated? First, through the autonomic nervous system. Second, through hormones secreted by enteroendocrine cells. These hormones include cholecystokinin, which is secreted by eye cells in the small intestine and promote enzyme secretion from the pancreas, and secretin, which is secreted by S cells in the small intestine and promotes bicarbonate secretion. I've added a list of other enteroendocrine cells found throughout the GIT. Now, what does a pancreas look like on slides? With a regular hematoxylin eosin stain, the Langerhans islets appear somewhat pale, while the small acini fills the other areas. Next, let's move on to the gallbladder. The type of epithelium in the gallbladder is simple columnar, and it's said to be the tallest columnar cells in the body. Beneath the epithelium, there's the lamina propria, which forms folds. There are no muscularis mucosae or submucosa. Instead, the muscularis comes directly after the lamina propria. This muscularis is important because it squeezes the gallbladder to empty it. Lastly, the outermost layer is the serosa for the peritoneal portion and the adventitia for the non-peritoneal portion. And one more thing. There are no mucus glands in the gallbladder because its epithelium can secrete mucin on its own. You might also find Rokitansky-Aschoff sinuses, which are deep invaginations of the mucosa that extend all the way into the muscularis layer. Muscle contraction in the gallbladder is stimulated by the hormone called cystokinin, which is secreted by eye cells in the small intestine. Now, let's take a closer look at the epithelial cells in the gallbladder. They are columnar, they have microvilli, and contain many mitochondria. The intercellular spaces are also relatively large, which is essential for concentrating bile by pumping out water. Now, let's talk about the biliary ducts. What do we mean by biliary ducts? They come from the liver and gallbladder and include the common hepatic duct, the cystic duct, and the common bile duct. The epithelium type in the smaller ducts is simple cuboidal, and in the larger ducts, it's simple columnar. These cells are called cholangiocytes. The small cholangiocytes tend to have microvilli, and the larger ones tend to have cilia on their apical surface. Below the epithelium, there's a lamina propria, followed by submucosa and muscularis layers. Unlike in the gallbladder, where there are no mucus glands, the biliary ducts have two types of glands. The saculae of Peale, a mucus gland, and the peribiliary glands, which are both mucus and serous glands. And that wraps up the key points about pancreas, gallbladder, and biliary ducts. I hope you found this interesting.